You're at the cutting edge of RNA therapeutics. I'm John Cook, professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist and director of our Center for RNA Therapeutics. Joining me in the studio is Dr. Christopher Brannon, my faculty colleague, who will introduce our guests.
Thanks, John. We're excited to have Dr. Jeff Collar here, who's the Bloomberg Distinguished Professor of RNA Biology and Therapeutics at Johns Hopkins University. Jeff has over 3 decades of pioneering research in mRNA stability and translation control mechanisms. He earned his PhD in cellular molecular biology from the University of Wisconsin, and then did postdoctoral research at Howard Hughes Medical Institute at the University of Arizona. And he's the former director of the RNA Center at Case Western University, where his group actually contributed to developing Spikevax COVID-19 vaccine. And beyond academia, he's now co-founder of Tevard Biosciences, which received Pfizer's Golden Ticket Award for Neuroscience Innovations. And he serves on the founding board and is a member of the Alliance for mRNA Medicines. So Jeff has over 6,000 citations and multiple RNA therapeutic patents, so he brings a huge amount of expertise to our discussion today on using post-transcriptional control regulation for novel genetic disease treatments. With that, I'll sort of kick off the discussion. I wanted to sort of get your opinion, Jeff, on what you see as major barriers that need to be overcome in the next 2 to 3 years for RNA therapeutics to realize some of their very huge potential.
Well, great. Thank you both for the invitation to be here today. It's a distinct pleasure to participate in this conversation. There's a lot of excitement about RNA-based therapeutics and what they can do, and the promise of what they can do. But I think one of the major limitations for getting to a point where we can realize these applications or these medicines for diverse applications is to get them to the right cells and the right tissue, as well as to improve their overall durability. And what I mean by that is how long they would actually last in the human body. And so it's, if we think about mRNAs, for example, mRNAs have proven themselves to be very successful in the use of prophylactic vaccine, because you can do an injection directly into the skin, and that causes an immune reaction that then has a memory response. But to take it to a point where we can use RNA for rare disease, we need to be able to access the organ that we need to affect, and we need to have the RNA last long enough that the patient actually sees some benefit. So I think those are some of the major sort of scientific challenges that we have in the next 10 to 15 years in order to realize this.
I think that's right, Jeff. Those are great points. The LNP, the lipid nanoparticles that we used for mRNA vaccines work brilliantly for delivery for vaccine. They're slightly inflammatory, which enhances their vaccine efficacy, but that's not very good for therapeutic protein delivery. If you want to deliver an RNA that is a protein replacement therapy, for example, it's not. What lipid nanoparticle developments are coming down the pike, do you see, that might help us?
I think because of the success of both Comirnaty and Spikevax, so the Pfizer and the Moderna vaccine, and both were delivered as LNPs, I think because these were successfully done and now are FDA approved, there's been a new reinvigoration into this space and how to develop novel LNPs that A, target the right tissue, and B, could be time released, for example. And there's a lot of work that has gone into this space to get to LNPs that could affect the brain, for example. Or maybe the better way to say it is that all LNPs easily go to the liver. Mm-hmm. And what a lot of efforts have undertaken in the last few years, based on what we've seen with the COVID vaccines, is how to move LNPs away from the liver, specifically detarget them from the liver so that they can get to other organs, and specifically to the pancreas or to the respiratory system, et cetera, the heart. And I think we're going to see a lot more of those come online very soon.
Wonderful. Well, we should probably get started, but before we do, perhaps you could introduce your young scientist who's joining us.
Yes, sorry. So joining us for discussion today is Dr. Ricardo Rivera, who's a postdoctoral fellow with my group working on some mRNA therapeutic designs, actually. So he'll be he'll be interrupting you here and there with questions, and yeah, we can get started with your presentation whenever you're ready.
All right, so I'll tell you a little bit of a short story of work that originated in my lab many years ago and is approaching sort of clinical applications. And this is work that has gone into founding of a specific company called Tevard Biosciences. But in general, my lab works on RNA metabolism, mRNA metabolism, and RNA metabolism in general as a way to develop precision therapeutics for various genetic disorders. And so just to start out, I have to have a conflict of interest statement. I'm a founder of Tevard Biosciences and a member of their SAB and have an interest in that company. So my lab works on mRNA and mRNA translation and stability. And the reason why we focus on understanding gene expression from the aspect of mRNA is that mRNA, of course, is the emissary of genetic information. It's what takes if you think of DNA as a restaurant, mRNA is the individual recipe for each dish that is made by the cell. And what's important to understand here is that you can actually stop making those recipes at any given time, and this is a critical aspect of gene expression. But mRNAs are actually read out in the cytoplasm. And so unless that recipe is essentially destroyed, it will continue to be read and turn into proteins over and over and over again. And so a critical aspect of gene regulation that my lab studies is actually understanding the timing events that lead to the downregulation of these mRNAs or these recipes so that they eventually turn off their expression in the cytoplasm. And we've studied this for well over 30 years about the relationship between the destruction of mRNAs and their relative translation. And we've now gone and used some of that information to treat a variety of genetic disorders that are called haploinsufficiency disorders or dosage disorders. And haploinsufficiencies are basically a class of diseases where there's usually a de novo mutation in one of the two copies of the DNA, either from mom or dad, that results in the loss of gene expression. So either the loss of the production of the mRNA or the loss of a functional protein. But the net effect is always the same, that where you need two copies of the protein in order to have a normal cell, you only have a single copy from either mom or dad. And this is manifest in what we call haploinsufficiency. And there are about 300 of these known in the human population. And they, for the most part, are very difficult to treat. Although from a theoretical standpoint, it seems like it should be relatively simple, that all we'd have to do is increase the overall expression of one of these two genes in order to ameliorate the phenotype. And so what we've gone after in terms of haploinsufficiencies is a variety of technologies that were the foundation for a company called Tevard Biosciences. And this is something that was founded between myself and Harvey Lodish, Daniel Fisher, and Warren Lambert. And the technology that I'm going to talk to you today about, which we use to treat not only certain classes of haploinsufficiencies, but also other genetic disorders, is what we call nonsense suppression, or tRNA suppressors. And in essence, about 20% of all genetic disorders are caused by the presence of a premature termination codon. And in the genetic code, of course, there are tRNAs that read the genetic code, and every once in a while you have a stop codon. And a stop codon is present in order to tell the ribosomes that read the mRNA that it's time to stop the translation of the genetic code and complete the synthesis of the polypeptide. But through mutation, what I'll talk a little bit about in a second, we can get what are called premature termination codons. And this results in a truncated polypeptide that is non-functional. And this is usually very devastating in terms of a genetic disorder because they're simply not only are they not making enough of the right protein, but sometimes they can be, in fact, creating a deleterious allele that could even have dominant negative effects. And a simple technology that really has been known about since the 60s is to recode tRNAs, these adapter molecules, such that they could read stop codons, and then insert an amino acid in the place of where that premature termination codon exists, and then result in the production of the normal polypeptide. So going back to our understanding of the genetic code, there are 64 words in the genetic code. 61 of those are encoding an amino acid, and 3 of those are designated as stop codons, and those are UAA, UAG, and UGA. And again, the important point here is that for these 3 stop codons, there is not a tRNA that exists within the human body. And so when the ribosome encounters those stop codons, it will stop making the polypeptide. Now, if we actually look at across all disease indications that are known within the human population, there are a lot of what are called nonsense mutations. So these are, again, the recoding of an amino acid, of a normal amino acid, to a stop codon. But it's not uniform across all of these other 61 codons. In fact, if you look at this, there's really only about 2 codons that are normally transformed into or mutated into a nonsense mutation or stop codon. And these are arginine to UGA and glutamine to TAG. And these account for better part of about 23 to 20% of each of those, so make up better part of 40% of all pathological nonsense mutations present within the population. And so when you look at this, you actually only need to make a handful of unique tRNAs in order to treat the vast majority of nonsense-related diseases. And the reason for this is based on some fundamental biology, which is the reason why RNA uses uracil and not, or sorry, I should say that DNA uses thymine rather than uracil that RNA uses. And this is because of this natural phenomenon, which is called cytosine deamination, where cytosine will spontaneously deaminate to uracil. And that basically is causing a mutation. And in DNA, the presence of uracil is actually sensed and then gets corrected whenever the spontaneous deamination occurs. And the interesting aspect of this is this deamination tends to occur more frequently when the C is next to a G. And so just going back one slide, you can see that this often can occur where this particular codon, CGA, arginine, which is a C next to a G, readily can be deaminated, and that turns into a U, which is, of course, a stop codon, UGA. And so that tends to be the most prevalent mutation you see in the human genome. So particular diseases tend to be enriched in nonsense mutations. One of those is Duchenne's muscular dystrophy. So about 15% of all DMD cases contain a nonsense mutation. But other muscular dystrophies, this can be as high as up to 42%. And so as a class of mutations, nonsense mutations make up the largest of all type of mutation that we see within human pathologies. And so having technology that can deal with these mutation-specific diseases, mutation-specific pathologies is really critical for treating these devastating disorders. And so we have developed a novel platform to create tRNAs that can read nonsense mutations. And this was done many years ago, in fact, first in my lab and then through Tevard, where we used technology to create suppressor tRNAs and then screen for tRNAs that would efficiently read through a bicistronic message, so a luciferase and then a nanoluciferase, and then look for appropriate restoration of the luciferase. And what you can see is through evolution of these tRNAs, we can get to tRNAs that will readily read through the stop codon mutation at a high frequency. And what's important here is then these experiments, we then deliver these suppressor tRNAs in the context of a viral vector, so AAV, in this case AAV9, and deliver it into a mouse. And what you can see, and this is really a first, where the model system for DMD, which is called the MDX mouse, produces no DMD or dystrophin. Dystrophin is the protein that is mutated in Duchenne's muscular dystrophy, and this MDX mouse has a mutation and is not making dystrophin at any appreciable level. But using various suppressors, you can see that we can get the restoration of dystrophin in these mice, and this really is a first, because dystrophin is one of the largest genes in the human body and very difficult to express and restore expression, especially. So being able to restore full-length expression is actually quite an achievement. In addition, using the AAV-based approach and injecting these intravenously into mice, we can see a dose-dependent effect on the restoration of dystrophin in various tissues, including heart and various muscles, and not as much as in the diaphragm. So again, consistent with the fact that we can get a systemic effect of the restoration of the dystrophin levels using the suppressor tRNA technology. And looking at this in more detail, you can see here, if we look at dystrophin by direct expression of dystrophin by a Western blot, there's none in the MDX mouse, and it's a wild type amount. And as we increase the overall concentration of the AAV vector, we get steadily higher increase in dystrophin expression in these mice, in this tissue, specifically the heart.
Jeff, quick question, and I'm sure you're going to address this, but the T suppressor would also suppress normal stop codons. What off-target effects might you see?
Yeah, that's a really good question, and I'm going to come to that in a second, John. And you would think that that's true, but it's actually not, and it's really surprising. And there's obviously some very cool biology here that needs to be considered. But let me probably have that slide. I have that slide in, I think, two more slides, and I'll address that question in a second. But just to drill on this a little bit more, if we look at polypeptides, so this is directly looking at polypeptides from dystrophin, before the PTC, the premature termination codon, and then after the premature termination codon, you can see in a wild-type mouse, you get peptides across the entire span of the gene. In the MDX mouse, of course, you don't see much of anything. But in a dose-dependent manner, when you express the suppressor, you get the restoration of these peptides. Now, I should note that if you're astute, you'll see that there's actually less peptides upstream of the PTC. And this is actually going to get a little bit at John's question, is because the presence of this premature termination codon elicits a quality control mechanism of that mRNA, which is called nonsense-mediated mRNA decay. And so this mRNA is normally a substrate for a quality control mechanism that recognizes that that mRNA harbors a nonsense mutation and needs to be destroyed. And this again goes to John's point, which is that the cell has an ability to tell whether or not a termination codon is normal or premature. And if it's premature, it's recognized and that mRNA is destroyed. And we don't understand all of the reasons why the cell can tell the difference between a premature termination codon and a normal, but it does. And in experiments that I'll show you in a second, you can see that the suppressor tRNA, in fact, never reads through the normal termination codon. It only reads through the premature termination codon. And again, this is just a quantification of these peptides following the premature termination codon. And you can see the increase in a dose-dependent manner. Okay, so to directly address John's question about reading the normal termination codon, if we look at this by peptides, that extended peptides beyond the normal termination codon. So we're looking for peptides that would exist within the 3' untranslated region of normal mRNAs. And what you can see is that across the genome, there's only 4 proteins where we can even see an extended 3' end of an mRNA, of the peptide, to suggest that there was any read-through of the normal termination codon. And when we quantitate that in more detail, in fact, the amount of read-through we're getting is really quite low. It's very negligible. And so this is, again, pointing to this fact that there's this natural system that exists to discriminate between normal and premature. And our suppressor tRNAs are not impeding that process and reading normal termination codons. The other side is that we can look at the tox of these suppressors, is looking at various assays of stress. And in the mice, you can see that when we look at these 4 different readouts of stress, there's in fact no real influence in a dose-dependent manner of the expression of the suppressor on the health of the mouse. And so this is, again, positive signs that these suppressors are safe and have very little toxicity, if any, even at high dose. Okay, so in looking in more detail at the mouse itself and looking at expression of dystrophin within various tissue, you can see that normally you can stain these filaments and they light up quite well in a wild-type mouse, where in the MDX-treated mouse, you've seen nothing. But in the MDX-treated mouse with the suppressors, we start to restore the normal localization pattern of dystrophin in a dose-dependent manner. And this is at 12 weeks following IV injection. And not only we can see that in various tissue, this is again this is the tibialis, and we see again the restoration of dystrophin levels within cell expression. And we can quantitate that and show that we're again getting a dose-dependent effect. Okay, so that's great. We can see that molecularly we're restoring dystrophin to higher and higher levels as we treat with these suppressors. How does that manifest into physiological outputs? And in mice, you can monitor the MDX phenotype using various strength assays, a rotor rod or a grip strength assay. And you can see that with these suppressors that we've developed, we are beginning to see an increase in the overall strength of the mouse consistent with the restoration of muscle function. I'd just like to point out, though, that these particular suppressors that I've shown you here, which we call TR0904, are in fact some of our weakest suppressors. They give us between 30 to 40% restoration of dystrophin levels. But we have 21%, sorry. But we have new generation suppressors that have not been tested yet in terms of mouse phenotypes that are in fact performing even better than these first and second generation suppressors that we've developed. And so we're very optimistic that as we continue to reiterate and evolve these tRNA suppressors, that we'll improve upon these phenotypes and get to a point where we can get even more significant physiological outcomes in the mouse model. So just to summarize, we've created tRNA suppressor technology and has the ability to effectively treat and possibly be an option for treatment of Duchenne's muscular dystrophy when caused by a nonsense mutation. And in addition to this, we think that this type of technology doesn't have to be just relegated to Duchenne's muscular dystrophy. It is a viable therapeutic platform for a range of disorders, including other neuromuscular disorders and cardiomyopathies that are associated with nonsense mutation. And I've shown you that the IV delivery of AAV expressing these second generation molecules can restore full-length dystrophin levels up to about 30%, and we see no observable adverse effects. In addition, the rescue of full-length dystrophin almost to 10% of wild-type levels seems to be associated with normal proteomic dysregulation and significant restoration of motor function within these mice, supporting the idea that it's working. And we've identified third and fourth generation, in fact, suppressors that have increased potency, some of which are giving us up to 50% expression of wild-type dystrophin in this mouse model. Wow. And so just with that, these are the team at Tevard that is responsible for the majority of this work. And if you're interested in the things that they do, you can go to their website here and learn more about the different disorders that we're trying to attack with the various technologies. And this is, of course, my lab, past and present members, and some of the people that give us money to do some of the work we do. And again, we're located at Johns Hopkins. And if you want to get in touch with us, this is how to reach out. So I'll stop there and take any questions.
Dr. Collar, I really enjoyed that. And I think it does just highlight how limitless the promise of RNA therapeutics can be. There's so many ways that one can modify the RNA and protein expression that is encoded by the RNA. And you've shown us very nicely another very intriguing way in which to modify translation of the RNA. I'm going to let my faculty colleague have the first shot.
Okay. Yeah, so I'm a little curious about how you think the suppressor tRNA is specific for premature termination codons. Have you thought mechanistically about what's going on there?
Yeah, I mean, we think about that a lot. In fact, because it's really perplexing, because it says that there's a lot of ways to think about it. And the fact that there is a mechanism, a natural mechanism within the cell to discriminate against these nonsense-mediated mRNA decay, says that the cell knows what it's doing. And it implies that there must be, at least in my mind, like a molecular ruler of some sort, which can tell the position of the termination codon, relative to something. And what that molecular ruler is, we don't really know. There has been some thought that that might have something to do with where exon junctions are within an mRNA. Others have thought that that might have to do with proximity of the termination codon to the 3' end. But it's a very active area of research. And we've actually been studying it in my own lab, and we've been looking at how the ribosome itself, through the protein that it makes, might influence the ability of the ribosome to recognize either a tRNA, a suppressor tRNA, or the proteinaceous factors that normally recognize a termination codon. But we really don't have the answer. But it's clearly there, and we just need to do more research to figure it out. And if we figure that out, then it opens up other opportunities to treat these nonsense, these disorders that are associated with nonsense mutations. And remember that up to about 20 to 25% of all human genetic disorders are the result of a nonsense mutation. And so if we have drugs or gene therapies such as this that can really go after that, there's a large number of patients that could really benefit from that primary research on understanding how the genetic code is really deciphered.
Yeah, I was thinking sort of about those metagene plots that you see when people map M6A modification genome-wide, and how there's always this peak right at the termination codon of that signal. And I was just wondering if maybe you think that might be a molecular marker for true termination codons as opposed to PTCs.
Yeah, and that's there's a great paper that showed that by Sami Jaffrey, and I think probably 10 years ago or so, that showed that peak near termination codons. And I'm pretty sure that Sammy's lab looked at that, and they, if I remember right, they didn't see that M6A were contributing to that. But that doesn't necessarily mean that it's not saying something else. That maybe the deposition of M6A around termination codons is the result of something that we don't completely understand. And there are certain things that we do observe through various experiments that are really interesting. So, for example, what you just talked about with this M6A seems to spike around termination codons, normal termination codons. But the ribosome itself, if we look at like ribosome profiling, where we can tell where a ribosome is sitting on an mRNA, it also tends to pause for a little while near a normal termination codon. And so that almost suggests that there really is a transition that's happening there. And remember that the ribosome is responding, in the case of deciphering the genetic code, it's responding to two things, either a tRNA or it's responding to a protein that recognizes the termination codon. And maybe it's that switch from recognizing a tRNA to recognizing a protein that is responsible for that pause or that M6A deposition, and understanding why that transition occurs might be the key to understanding why you can suppress at a nonsense mutation but not at a normal termination codon. Yeah. But that's all just, hand waving.
Yeah,
what would happen if it...
Go ahead. Sorry.
No, you could.
I guess one last
ahead.
question, and then I'll turn it over to Ricardo. But yeah,
Sure.
I know people have been using compounds that actually inhibit nonsense-mediated decay, and those kind of operate in a similar fashion where they seem to be specific to PTC read-through. Have you ever tried coupling any of those compounds with your suppressor tRNAs to see if they?
I never have. Yeah, that'd be a really fun thing to do. Because the challenge you always have with the nonsense mutation is that you have competition with, reading the premature termination codon by the suppressor, and then that template being destroyed by nonsense-mediated decay. And so every time a ribosome experiences that premature termination codon, it has an option. And that option is either to find that suppressor tRNA or not find it and then be destroyed. And if it's destroyed, then there's no opportunity to reread it. And so there is the potential to improve the overall efficacy of the suppressor by eliminating nonsense-mediated decay. So it would be interesting to test that. But from a therapeutic development standpoint, we haven't done that because these sort of combinatorial approaches are difficult to do through FDA trials and such.
Right. Ricardo, you had a question?
Yeah, I was going to ask if have you observed that same trend with other genes with nonsense-mediated nonsense mutations as well?
Yeah. So in fact, when we have, in fact, so we've looked at other, like I said, every disease has nonsense mutations associated with them, and some of the other indications we work on are cardiomyopathy associated with Titin, which is another incredibly large gene in the human genome. And we see very similar types of results with Titin as we do with dystrophin. And we've also had some successes of dealing with this haploinsufficiencies in the brain. But the DMD program and the cardiomyopathy program are the ones that are the most developed. But to be honest with you, it goes back to kind of the conversation that we had at the beginning, which is about delivery, because it's all delivery. And then when it comes to the RNA space, it doesn't matter whether you're talking about a tRNA like I am, or you're talking about an mRNA, or you're talking about a small RNA therapeutic. You got to get it to the right spot, and that delivery always seems to be a challenge. Right. So in the laboratory, you can get these suppressors to read through almost any gene, get it to read through a premature termination codon. Doesn't matter what it is. Right. But if you want to get it to the brain, or if you want to get it to the liver or the pancreas or wherever, the lungs, that's where you're limited more than anything. It's the delivery mechanism.
You showed us some nice data with grip strength with the DMD, the T suppressor therapy for the animals with Duchenne muscular dystrophy. What about the heart? Did you see any benefits in the cardiomyopathy that occurs in those animals?
Yeah, we haven't looked at that yet because we're trying to get the third and fourth generation tRNAs, and those are studies that are ongoing to get more. Our first readouts were to look at grip strength and these other phenotypes that are classically associated with DMD. But if you remember in some of the slides, I could see that I showed, we are seeing improved dystrophin levels in the heart. And so reading out heart function will be something we can do as we move forward.
If you need some help with the functional evaluation of the heart, we're here, happy to help.
I know, John. Thank you very much. We'll give you a call.
Well, Jeff, thanks so much for a beautiful presentation. Dr. Brannon, Ricardo, thank you for joining in the studio, and just a wonderful talk. So we'll bid adieu, and you have been joining us at the cutting edge of RNA therapeutics today, where our host, Dr. Jeff Collar, has talked to us about his beautiful data with T suppressor therapy for DMD. Thanks again, Jeff.